Good morning again, everyone. I will present to you how we run this extensive project across Madagascar. If today we know the amazing biodiversity of Madagascar, that is thanks to the scientists who described them for us. We will not have any species to tell to tourists without the scientists. And as Fredrik already told, many of the well-known biodiversity are vertebrates and plants. So this is why IBA came into a very helpful project to help us to know more about the very small fraction of insect that we don't have the knowledge. So how did we start IBA? First, we considered all the habitats and ecoregion, and we discussed also with other scientists about any special habitats that we should take into account. Okay. Second, we analyzed satellite images to make sure that forest is still there, because sometimes we know that there's a park, but sometimes there's no forest anymore. Sometimes the border of the forest has changed. So that's why we check the satellite images. And then we analyze also the topography to make sure that we can get to that site and from where we should go to reach the sites. Of course, we approached the park managers to inquire about any special issues locally and to know which assistance and how to work with the local people. And besides this, of course, there are the security aspect and the village, which village are next to the site. And we rejected the sites that has no village around it. Otherwise, it would be impossible to run the project. So these are the sites that we kept to work. So we have 50 traps distributed at 32 sites, and I take the opportunity to thank again the PBZT and the Ministry of Ecology, and of course all the park managers. So these are the sites that we chose, and if you notice, there are some places where we don't have sites because it's not possible to go, and even there's no village around and it's too far. First, we need to go to the sites, and it took us 90 days to just install the traps and train the assistants. And as you see, there are many, even for the start, it's already a challenge, and we learned from the installation process that how it would be later on and what kind of challenge we would face when we will run the project. And as usual, before we install the trap, we meet the local chiefs of the village and you explain them about the project, because this is part of the Nagoya Protocol. They need to know what we are doing for the project, and that we are collecting the specimens only for identification. There's no commercial purpose behind it. And this is very important because later on, if it's not explained, then Madagascar is a country of rumors. People would say that we are selling insect or whatever. And we are working with the park manager again to choose the assistants. They help us by providing the assistants. And when we are in the forest, we are looking for flat and a bit open place to install the trap. So we don't open new area, we don't clear new forest, we don't clear the new places to set the traps. And in total, we've been working with hundreds of assistants from the south to the north. And we started the sampling in the first week of August 2019. Because during the 90 days, we trained, we installed the traps. and we didn't sample yet. So during those days, during the 90 days, some traps, some sites had the opportunity to train themselves for collecting the data. So the sampling lasted on exactly 1 year. On our side, we split into 2 teams to follow up the sites. So we have the south sites and the north sites. And Tsir was coordinating the south sites, when I was coordinating the north sites. Because the 100 assistants, we need to call them every week to make sure they're working. So the assistant, they collected the sample weekly, and they used an application on smartphone on which they scan the codes, and these codes and those data, they are uploaded on website that we check. So every week we check like who didn't work or who worked correctly. And for your information, the application has GPS, so it recorded the GPS coordinating when they are working. So this for us to make sure that they've been working at the traps but not at the village. And every 2 weeks or 3 weeks, there's a local supervisor who checks the data. Because sometimes that's one challenge in Madagascar, then the network doesn't work. So they couldn't upload the data, but they wrote down the data on a notebook, and the local supervisor checked those samples against the data in the notebook. And every 3 months, we went to all sites to check the traps and collect the samples. And during that time, we also collect the PV de constatation and the transport permit. And many of you have noticed that when you work in Madagascar, I don't have experience abroad, but here in Madagascar, administrative paperwork is really part of the research. And it costs time and effort. It really costs time and effort, but we all need to comply to the rules. So we make report to the Malagasy institution and the park managers, and every 6 months we have to renew the research permit. Every time we go to the sites, we meet the park director and we report to them. And we take also that opportunity to renew the field item and the field materials. And by the end of the sampling time, we prepared the export permit. And this was the one challenge. This was during the COVID. It was really during the COVID problems. So during the lockdown, sometimes we had like, how can we check? How can we move around? 70%, 75% of the samples was sent to Sweden, and 25% stays at MBC and PBZT. This is part of the rule in Madagascar. And just as a reminder, all the samples have been sequenced, and the DNA analysis, we start the talk, is still ongoing. And the 25% of the samples in Madagascar will be used to create the reference library, which is the most important part, to give name to the species. When we collect the samples, when it arrives at MBC, we check the samples, and we check again the data from the website before we store them. To explain the temporal and spatial distribution of the species, we collected ecological parameters. First, we measured the site characteristic and also the ecosystem functions. To measure the daily temperature and humidity, we use a data logger, a very efficient one because you just live in the forest. Of course, you need to cover it against rain, but it's very easy to use. You just put the battery and you put it in the forest, and it runs for 1 year. So these are the parameters that we measured. We measured the humidity of the soil, and we collected soils that we sent then to Laboratoire de Radioisotope for chemistry analysis. And we also sampled for arthropod in the litter using a sifted litter. And we characterized the habitat, the forest habitat, by measuring the DBH and the sapling trees of each site. And to study the canopy, we take picture of the canopy using the same smartphone at the same angle. And this was used to measure the canopy opening. For the soils, we use the lamina bait. So this is a plastic with holes and filled with one with beans and one with eucalyptus. And we put it in the soil, first one for 21 days, and the second one, the tea bag, for 60 days. So these are used to study the decomposition activities of the microorganisms in the soils. After that time, we count how many of these have been eaten by the microorganism in the soil. And after that time, after the 60 days, we took back the tea bag and brought it to Laboratoire de Radioisotope, and they weighed it, and they knew how much have been eaten by the microorganisms in the soil. For the functions of the insect communities, we studied herbivory, seed dispersal, and predation. So for the herbivory, we picked up in total 100 leaves across 4 transects, and we take the picture. So the picture has been taken in the same condition, same distance, same, and there's the scale. To study the seed dispersal, like what is the preference of the insect, we put some seeds and we counted the number of seeds that has been taken by insect. To measure the predation, we used mealworms and tuna. And again, we count the number of mealworms left on the dish, and for the sardines, we weight the remaining sardines on the dish. So we counted them the first hour, and then we counted them again the next day. So what are the next steps? As again, all the samples have been sequenced, but there's still this process of matching and identification. We still need Malagasy researchers to collaborate with, and the publication is also ongoing. All the data, all the sequence data will be publicly available on GBIF. So this is the most important aspect, that all data will be later on available to anyone. Thank you.
Thank you, Dimby. We have time for one question.